The garden industry has a ready answer for anything that costs $1 and cannot be bottled. They call it a myth. They called it that about this method too. Then someone kept a logbook. 2 identical cucumber plants, same seeds, same soil, same water, same sunlight, same measured care, except for one thing. On day 7, one plant received a homemade brew made from a $1 packet of baker's yeast and a spoonful of sugar. The other received plain water. By day 28, one plant stood 40 inches tall, thick as a broom handle, heavy with blooms. The other was barely knee-high, thin-stemmed, and yellowing. At harvest, the control plant produced 3 cucumbers weighing just under a pound. The yeast-fed plant produced 9, firm, plump, and heavy, totaling 3.1 pounds, more than triple the yield. 1 ingredient, 1 watering. Numbers logged in ink on July 12th. That is not a claim. That is a record. When those numbers started circulating, mainstream horticulture had its ready response. Anecdotal, not reproducible under controlled conditions. The kind of result that happens in someone's backyard story, but falls apart the moment you put a scientist in the room. Then scientists put themselves in the room, controlled greenhouse trials, wheat and sugar beet, measured root biomass, documented yield data, and the numbers held. This is the story of a biological method that costs less than $1, has been practiced across multiple continents for centuries, is backed by documented data, and has never appeared on a single garden center shelf. Not because it doesn't work, but because living organisms cannot sit in a bottle waiting to be sold. Welcome to Roots Conspiracy. The garden industry has a very specific word for anything that costs nothing and cannot be sold. They call it exaggerated. They called it that about yeast water too. Then the greenhouse data came back. And here is why that pattern keeps repeating. The fertilizer aisle at your garden center is not organized around what works best for your plants. It is organized around what survives the journey from factory to warehouse to store shelf to your cart. Those are 2 completely different design criteria. And the industry has chosen the second one every single time. Walk into any garden center and look at the fertilizer section. Dozens of products, dozens of formulations, NPK ratios, chelated micronutrients, mycorrhizal inoculants in sealed packets, slow-release polymer coatings. The messaging is consistent across all of it. Your soil is complicated, your plants have complex needs, and you need the right product, their product, to address them. Don't improvise. Don't experiment. Buy the solution. Against that backdrop, baker's yeast and sugar sounds like a folk remedy, something that belongs alongside moon planting and talking to your tomatoes in the category of gardening myths. That is exactly the category the industry wants it in. Because here's what that fertilizer aisle does not tell you. The products on those shelves were not designed to maximize your plant's health. They were designed to maximize shelf life. A bag of blue crystal fertilizer can sit in a warehouse for 2 years and deliver exactly the same performance on day 730 as it did on day 1. That stability is not a feature for your plants. It is a feature for the supply chain, the living biology that actually drives root growth, the enzymes, the auxin-like compounds, the B vitamins that yeast produces during active fermentation cannot survive that journey. It has a working life measured in hours. No preservative can stop fermentation without killing the yeast. No packaging maintains biological activity across weeks of shipping and shelf time. So the industry does not sell it. And because the industry does not sell it, it does not fund research on it. And because research is not funded, mainstream horticulture has limited peer-reviewed data on it. And because peer-reviewed data is limited, experts call it exaggerated. That is a circular system that has nothing to do with whether it works. It has everything to do with whether it can be monetized. But this method is not new, and it did not start in someone's backyard. In the hills of South Korea, farmers have relied for generations on fermented plant juice, FPJ, crafted by packing wild greens with brown sugar and letting microbes work in a shaded jar for a week or more. The resulting liquid, alive with yeasts and bacteria, gets applied to fields to spark growth and resilience. The Jadam natural farming movement simplifies this further with rice wash water fermented with sugar, building a potent yeast culture in days. In English cottage gardens, comfrey teas fermented with a sprinkle of sugar have been poured around tomatoes, cucumbers, and beans for centuries. Even in Soviet-era dacha gardens, handwritten manuals described yeasty ferments for fast, cheap plant feeding when commercial fertilizers were out of reach. From the rice paddies of Korea to the allotments of England, across centuries and continents, gardeners independently arrived at the same conclusion. Living, bubbling ferments do something bottled chemicals cannot. They activate the soil, feed roots directly, and set off a chain reaction of growth. The industry calls it exaggerated because the industry cannot sell it. That is the only reason this is a debate at all. To understand why this works, you need to understand what baker's yeast actually is and what it produces when it wakes up. Inside every packet of dry baker's yeast, Saccharomyces cerevisiae, the same organism that makes bread rise, are millions of living cells in a dormant state. When those cells encounter warm water and sugar, they activate and begin consuming the sugar through fermentation. That fermentation process is not just producing carbon dioxide bubbles. It is generating a cascade of biologically active compounds that plant root systems have been responding to for millions of years. Dr. Anika Nguyen, a plant physiologist at the University of Minnesota, traces the mechanism directly. During active fermentation, yeast cells produce cytokinins and auxins, plant hormones that trigger cell division and root elongation. When roots absorb these compounds, they behave as if given a direct signal to expand and branch, drawing in more water and minerals from the surrounding soil. The result is a root system that grows deeper and denser, supporting taller stems and larger leaf canopy above ground. Alongside these hormone-like signals, fermenting yeast releases B vitamins, particularly B1, B6, and B12, along with amino acids and enzymes that function as direct building blocks for cellular growth. These are not synthetic approximations of plant nutrients. These are the actual organic compounds that exist in healthy living soil, produced by living organisms, in a form that root cells recognize and respond to immediately. Dr. Mateo Alvarez, a soil microbiologist, identifies a second mechanism, nutrient unlocking. As yeast cells multiply and then die off, their cell walls break down, releasing phosphorus and nitrogen in forms that plant roots can absorb directly. In laboratory trials, wheat and sugar beet treated with yeast-based solutions showed 20 to 30% higher yields compared to untreated controls. The effect is strongest in soils low in available nutrients, where yeast acts as a living bridge between locked-up minerals and hungry roots. There is a third mechanism that mainstream fertilizer marketing never mentions. By colonizing the root zone, Saccharomyces outcompetes many harmful bacteria and fungi, forming a protective barrier that blocks disease-causing invaders. This probiotic effect is documented in greenhouse studies where plants treated with yeast fermentations show fewer root diseases and recover faster from stress. Yeast activity in the rhizosphere also helps plants regulate water loss, making them more resilient to drought and heat. Now compare all of that to what a bag of blue crystal fertilizer does. It delivers salts. Your roots absorb those salts passively through osmosis. There is no signaling. There is no activation of the plant's own internal growth systems. There is no probiotic protection. There is no nutrient unlocking. You are feeding the plant from the outside. You are not turning on what is already inside it. Yeast water activates the plant. Chemical fertilizer supplies it. Those are fundamentally different mechanisms, and the one that activates costs $1.30. If this is the kind of content you've been looking for, hit subscribe. Roots Conspiracy covers practical methods backed by real data that somehow never make it into mainstream gardening advice. Now let's get into exactly how to make this and what to watch for when you apply it. Here is the recipe that no garden center will ever put on a display. Not because it's complicated, because it's alive. What you need: 1 liter of lukewarm water, 2 tablespoons of dry baker's yeast, approximately 20 grams, 1 tablespoon of granulated sugar, a clean jar or container, a cloth or loose lid. Total cost: less than $1. Water temperature is the first critical variable. Lukewarm means comfortable to the touch, around body temperature, 35 to 38 degrees Celsius. Too hot above 45 degrees kills the yeast cells instantly. You will have a dead brew that does nothing. Too cold, and the yeast stays dormant and fermentation never starts. This single variable determines whether the brew is alive or wasted. Step 1. Pour 1 liter of lukewarm water into your clean container. Step 2. Sprinkle 2 tablespoons of dry baker's yeast into the water. Even an expired packet from the back of the drawer will often work. Try it before buying new. Step 3. Add 1 tablespoon of sugar. The sugar is not for the plant. It is pure fuel for the yeast. Without it, fermentation does not start. With it, the yeast begins waking up within minutes. Step 4. Stir thoroughly until both yeast and sugar are fully dissolved. Every particle needs contact with the warm water to activate evenly. Step 5. Cover with a cloth or leave a lid slightly ajar, never sealed. Sealed containers are dangerous. As fermentation produces carbon dioxide, pressure builds fast inside a closed container. Plastic bottles bulge and deform. Glass bottles can shatter. Even a screw cap can pop with force. Always leave an escape route for the gas. Keep the container away from direct sunlight. UV disrupts fermentation. Leave it at room temperature for a minimum of 2 hours, ideally up to 12 hours for maximum biological activity. What you are watching for: foam. A visible layer of froth on the surface, small bubbles rising and accumulating like the head on a freshly poured beer. This is carbon dioxide being released as the yeast consumes the sugar. It is your confirmation that the brew is alive and active. No foam after 2 hours means something went wrong. Check the water temperature, check the yeast age, and try again. Yeast packets are cheap. Step 6. Dilution. This is not optional and cannot be skipped. Take the entire 1 liter of fermented concentrate and add it to a 5-gallon bucket of clean water. This creates the correct dilution, strong enough to deliver the full biological payload, gentle enough to avoid burning roots. Applying the concentrate directly to plants is a fast way to cause damage. The active compounds at full concentration can overwhelm young root hairs, causing wilting, browning, or complete stalling of growth. The diluted solution is gentle enough for seedlings and strong enough for mature plants. Always dilute. Always. Step 7. Pour the diluted solution directly around the base of each plant, soaking the root zone thoroughly. Do not spray the leaves. The target is underground. Stir the bucket well before each pour to distribute the biology evenly. Timing and frequency. Use the solution within a few hours of brewing while the yeast is still active and the signals are fresh. Discard any leftover. Do not store it. Do not seal it. Do not save it for tomorrow. Apply every 7 to 14 days. More frequent applications do not increase the effect. They risk tipping the balance, particularly in young plants or containers. The reason it cannot be stored is the same reason it cannot be sold. It is alive. And a supply chain built on dead chemistry has no room for anything alive. That gap between what works and what ships is not an accident. It is the business model. Here is what the industry cannot explain away. A logbook, dated entries, measured outcomes, 2 identical plants, 1 variable. The entries begin on a cool morning in early May. 2 identical seeds dropped into fresh soil side by side. Same sunlight, same water, same measured care, except for a single watering on day 7. 1 plant received 1 quart of homemade yeast brew poured at the base. The other received plain water. By day 12, the difference was already visible. The control plant stood just 8 inches tall, leaves tinged with yellow, edges curling. The yeast-fed plant had surged past a foot, new leaves unfurling with a rich, glossy green. The gardener's note in the margin, growing like mad. Never seen anything like it. The daily entries read like a race that was already decided. Day 15. The yeast-fed plant stretched to 18 inches and was sending out side shoots. Day 20. First flowers appeared. Day 28. The control plant was barely knee-high, stems thin, while the yeast-fed plant stood at 40 inches, thick as a broom handle and heavy with blooms. The logbook records the moment the gardener was forced to add a second stake just to keep the vine upright. When harvest arrived, the scale told the final story. The control plant produced 3 cucumbers, together weighing just under a pound. The yeast-fed plant yielded 9, each plump, firm, and heavy, totaling 3.1 pounds. Fruit count and weight logged in ink alongside the date, July 12th. 1 watering, 1 ingredient. A yield more than triple what the control produced. The numbers in black and white. Not a claim, a record. Now here is what healthy plant response looks like in the first 72 hours, so you know exactly what to watch for in your own garden. At the soil surface, fine white feeder roots become visible. The plant is actively extending its root network into the area where the brew was applied. The leaves develop a noticeably deeper green as the plant begins accessing increased mineral uptake through its expanded root zone. The stem shows increased turgor, standing straighter, firmer, with more structural integrity. In some cases, a faint sweet smell lingers around the base for 24 to 48 hours while the yeast remains active in the soil. Signs that something went wrong are equally recognizable. If the brew was applied undiluted or too frequently, root tips will brown. Leaves may wilt within 24 hours. The plant looks worse, not better, in the first 2 days. If this happens, flush the root zone with plain water immediately and wait at least a week before reapplying. The most consistent results appear in 3 situations: plants in depleted or nutrient-poor soil, fast-growing vegetable crops like tomatoes, beans, cucumbers, and peppers, and plants already under stress that have pushed their root systems to their current limits. In these situations, the auxin-like signals hit a plant primed to respond. The root system expands rapidly. Mineral and water uptake increases measurably. The above-ground plant responds within days with visible signs of recovery and accelerated growth. The logbook is one data point. The greenhouse trials are another. Together they describe a consistent pattern, one that the fertilizer aisle has never had a reason to show you. Here is the straightforward version of why this method remains controversial. It cannot be sold. Not won't be sold. Cannot be sold. The moment yeast meets sugar and water, it launches into active fermentation, multiplying and releasing carbon dioxide continuously. In a sealed bottle, that gas has nowhere to go. Plastic bottles bulge and deform within 24 to 48 hours. Glass bottles can shatter. Even vented caps cannot keep up. And as the yeast consumes all available sugar, the solution turns sour, the biology collapses, and what remains is a flat, spoiled liquid with none of the active compounds that made it work. Commercial fertilizers avoid this entire problem by selling salts, dead minerals that sit inert on a shelf for years. No gas, no spoilage, no logistics nightmare, no liability from exploding bottles. That stability is what makes the commercial model work. A 5-pound bag of name-brand fertilizer runs $30 to $50. Concentrated biostimulant bottles fetch even more. The ingredients for a home-brewed yeast solution cost less than $1. There is no patent on Baker's yeast and sugar. There is no proprietary blend. There is no margin. Garden supply companies cannot profit from something so simple, so perishable, and so easy to replicate at home. To put live yeast water on a store shelf would require constant refrigeration, vented packaging, rapid turnover, and constant quality control, all driving up costs while shrinking margins to nothing. The industry sticks to what it can control, and it calls everything it cannot control anecdotal. That is not a conspiracy with meetings and memos. It is a structural outcome. The profit equation simply does not favor living biology. But there is a second reason experts push back, and this one is worth taking seriously because it is legitimate. The results are genuinely inconsistent across different conditions. In already fertile, loamy soil packed with organic matter, yeast water provides minimal additional benefit. The plant already has easy access to the signals and nutrients the brew is delivering. Adding more does not move the needle significantly. In heavy clay soil that restricts root growth physically, no amount of biological signaling overcomes the mechanical barrier. In overwatered conditions, live yeast can contribute to mold or unwanted microbial activity in the root zone. The 30% root biomass gain that gets cited is real, but it is the high end of the range, measured in nutrient-poor sandy soil with crops that respond aggressively to auxin signals. The average gain across all conditions is lower. Some gardens see dramatic results, others see nothing noticeable. The inconsistency is real and worth knowing. The experts who call this exaggerated are not entirely wrong about the inconsistency. They are wrong about the mechanism. The underlying biology is solid and documented. What is exaggerated is the claim that it works identically in every soil for every plant. What is not exaggerated is what it does when conditions are right. And the conditions where it works best are exactly the conditions where most struggling gardeners find themselves. Depleted soil, stressed plants, and a fertilizer budget that is already stretched. Chemical fertilizer sales top $200 billion a year globally. The barrier to living alternatives is not biology. It is habit, industry inertia, and a supply chain that has no room for anything it cannot bottle and ship. Here is the part that mainstream gardening sources will never say out loud. This method has real limitations, and it still outperforms what they are selling you under the right conditions. Baker's yeast water is not the universal plant booster that some gardening communities claim it is. It works best in specific situations. It requires fresh preparation every single time with no exceptions. It demands precise dilution, or it causes the exact damage it is supposed to prevent. Results vary enough across soil types and plant species that no honest claim can guarantee it will work in your specific garden. Those limitations are real. Acknowledge them. Test before scaling. Start with one plant, watch for a week, then expand if growth responds without signs of stress. But here is what those same mainstream sources do not say. The biological mechanism is real and documented. Auxin-like compounds from yeast fermentation trigger root cell division. B vitamins and enzymes from active yeast fuel cellular growth at a level that dead chemical salts cannot replicate. Cytokinins trigger cell division above ground. The probiotic effect protects the root zone from pathogens. Controlled field trials in wheat and sugar beet showed 20 to 30% yield increases compared to untreated controls, repeatable across different soils and climates. The gardener's logbook showed 3.1 pounds versus 9 pounds from identical plants with one variable changed. Those numbers were logged in ink on July 12th. They are not a claim. They are a record. Your job is to know where this works and apply it there. Depleted soil, fast-growing vegetables, stressed plants, fresh brew every time, correct dilution, 10 parts water to 1 part concentrate. Root zone application only. Use within hours. Apply every 7 to 14 days. Under those conditions, a $1 packet of Baker's yeast and a spoonful of sugar may do more for your root zone in 72 hours than anything on that fertilizer shelf has done all season. And here is the question that this channel keeps asking. The method is documented. The biology is understood. The data is real. The historical practice spans Korea, England, Russia, and dozens of other cultures across centuries. It costs less than $1. It takes 30 minutes. So why is this the first time most gardeners are hearing about it in these terms? Why has no extension office built a program around it? Why has no gardening magazine run a cover story? Why does the fertilizer aisle have 47 products and not one of them is alive? The answer is the same answer it always is. There is no profitable way to sell it, so the industry ignores it. And because the industry ignores it, the media ignores it. And because the media ignores it, most gardeners never find out. That is not an exaggeration. That is the pattern. And it does not stop with yeast water. The pattern is always the same. The knowledge exists. The data exists. The historical practice spans centuries and continents. What does not exist is a profitable way to package it, ship it, and put it on a shelf with a margin that justifies the logistics. So the system delivers you something else instead, something dead, stable, and expensive, and calls this one exaggerated. That is not a coincidence. That is a business model, and it is the same business model this channel keeps documenting, not just with yeast water, but across every method, every ingredient, and every technique that works, costs nothing, and cannot be sold. That is what Roots Conspiracy is built around, not just this method. The broader pattern of what gets sold, what gets dismissed, what gets forgotten, and who benefits from each outcome. If that conversation is worth having, subscribe and stay close. The next video goes deeper. We'll see you there.